First question, preparation of recombinant DNA. Who wants answer? Please answer the question. Who wants answer the question? Preparation of recombinant DNA describes the methods of making recombinant DNA.
Can I answer?
Okay, answer the question.
And the first questions, preparation of recombinant DNA.
Okay.
Recombinant DNA is rDNA, molecules rDNA, and is a molecules formed by laboratory methods of genetic recombination, such as molecular cloning, and to bring together genetic material from multiple sources and creating sequences that would not otherwise be found in the genome. Recombinant DNA is a general name for a piece of DNA that has been created by combining at least two strands. Recombinant DNA is a possible because the DNA molecules from all organisms share the same chemical structure and differ only the part of the genomic actually because is the molecular biology and the biomedicine. Recombinant DNA molecules are sometimes called chimeric DNA because they can be made of material from two different species and like the mythical chimera. And to make DNA technology uses palindomic sequences, and the recombinant DNA is made by combining DNA from two or more sources in a genome.
Zamira Karibay. Okay, good. Very good. The next question describes the methods of making recombinant DNA. Who wants to answer?
Can I answer, teacher?
Okay.
Recombinant DNA is the methods of joining two or more DNA molecules to create a hybrid. The technology is made possible by two types of restriction endonucleosis and liase. A restriction endonuclease recognizes a specific sequence of DNA and fits within or close to that.
Okay.
That also.
Mirano. Mulder, Sirikwayo. Answer the question. Who wants to answer the question? Okay. Continue our lesson. Seminar work 13. DNA technology in the medicine, pharmaceutical, and the food industry. The aim of the work studies the use of DNA technology in the pharmaceutical, medicine, and the food industry. The importance of the DNA technology. Recombinant DNA technology is a major DNA-based tool that opens a new age for modern biotechnology. With this technology, a gene or multiple genes can be identified, cut, and inserted into the genome of another organism. In 1972, Stanley Cohen and Herbert Boyer developed recombinant DNA technology. The restriction enzymes play an important role in this technology. The restriction enzymes will cut at a specific site within the DNA molecule called a restriction site. The most commonly used vector and plasmids are bacteriophages. Besides, vectors should also carry sequences for selection and identification, such as gene for ampicillin resistance and the cloning sites. To date, host organism includes bacteria, fungi, and animal cells. To introduce vectors into hosts, techniques involving microinjection, biolistics, gene gun, alternate cooling and heating, and calcium phosphate ions has been used. Recombinant DNA technology has 5 steps. First step, cutting the desired DNA by restriction sites from any organisms, and cutting the vectors by the same restriction enzymes, and amplifying the gene copies by PCR, and inserting these genes of interest into the vectors, and obtains in the result obtains the recombinant DNA vectors, recombinant vectors. First step in transferring these recombinant vectors into host organisms. This process called transformation. The recombinant DNA containing target genes transformed into host cell. And then obtaining then first amplification and multiply these genes in host cells and obtaining the product of recombinant genes. DNA technology in the pharmaceutical industry. Recombinant DNA technology has resulted in the generation of over 150 recombinant therapeutic proteins that are approved for the treatment of human diseases, more than half of which are produced in cultivated mammalian cells. The first recombinant proteins included beta interferons and the tissue type plasminogen activator TPA. The later became the first FDA approved recombinant therapeutic protein activates in 1986. FDA at the Food and Drug Administration means FDA. Recombinant DNA technology in the medicine. Recombinant DNA technology has been widely used in the diagnosis of diseases using recombinant proteins, in the detection of normal genetic variation, in the detection of genetic variation causing diseases, and in prenatal screening for genetic diseases. DNA technology in the medicine. Continue using this technology, the first golden molecules of the medical biotechnology were produced, namely human insulin. The synthetic human insulin was produced by use of recombinant DNA technology. Nowadays, millions of diabetics worldwide use synthetic human insulin to regulate their blood sugar levels. The synthetic human insulin is made in both bacteria and yeasts. Recombinant DNA technology applications in clinical medicine include the test for the genes causing sickle cell disease, Huntington disease, and dozens of other Mendelian disorders. Recombinant DNA technology enables the manufacture of proteins and antibiotics with a defined specificity and uniformity. In medicine, recombinant DNA techniques can be used for First, the diagnosis of disease and the investigation of their pathogenesis, and the second, the detection of carriers of genetic diseases. Third, the large-scale synthesis of medically important peptides which would otherwise be in short supply, such as human insulin, growth hormone, and interferon. In addition, DNA technology may ultimately be of value in gene therapy in cases where a disorder is attributable for a defective gene. DNA technology in food industry. Increase in quality of plant products. Golden rice containing beta carotene to overcome vitamin A deficiency in a region where rice is a staple food. Canola containing high levels of oleic acid. Tomatoes which do not rot in room temperature. Other vegetables and fruits with delayed ripening as well as modified flavor characteristics. Control questions describe the importance of DNA technology in pharmaceutical industry, DNA technology in the medicine, and the DNA technology in the food industry. Answer this question in next lecture. Prepare the presentation material, DNA technology in the pharmaceutical, medicine, and the food industry, and send me by university system. Watch the video, how synthetic insulin is made using recombinant DNA technology from bacteria.
Synthetic human insulin was the first golden molecule of the biotech industry and the direct result of recombinant DNA technology. Currently, millions of diabetics worldwide use synthetic insulin to regulate their blood sugar levels. Synthetic insulin is made in both bacteria and yeast. In order to get bacteria to make insulin, a number of factors need to be considered. Genes in eukaryotes have introns. Bacteria do not have introns in their genes, and so they do not have biochemical machinery to remove introns. Also, eukaryotic proteins are processed after translation. Insulin is first translated into preproinsulin, which is 108 amino acid long. First 24 amino acids are signal protein, which leads preproinsulin out of cell. It is cleaved off, leaving proinsulin in pancreas for further processing. Protein folds into looped structure as disulfide bridges are made between cysteine amino acids. 33 stretch of amino acid is cleaved off, leaving mature insulin. In order to get bacteria to make insulin, first DNA is made based on protein sequence of 2 insulin chains. A and B. These DNA fragments are inserted into plasmids. Each DNA fragment is inserted into the beta-galactosidase gene on a plasmid. The plasmids also have tetracycline resistance gene. Plasmids are then transformed into bacteria. Tetracycline is added to kill off any untransformed bacteria. The transformed bacteria are grown. Then beta-galactosidase and insulin fusion protein is harvested and purified. The beta-galactosidase part of the protein is cleaved off and discarded. The 2 protein chains are mixed finally into insulin, made from bacteria.